Okay, so I'll just like someone to come up to draw the cell cycle on the board. Then just see if you how much you know, yeah, before we move on to the next part. Who was not here for Miss Liu's lesson? OGLs, is it? Okay. What? Okay. Alright, the cell cycle. Okay, ready? Let's see what. Alright, Amanda was not here for lessons. Michelle, will you draw the cell cycle on the board for me? Yeah. As Michelle is drawing a cell cycle, could everyone also be attentive? I mean, just swap you out, swap you in at any time. Huh? Okay. The cell cycle. What is the cell cycle? What does it look like? Okay, she knows there's 4 stages. Are there 4 stages? Okay, G1, S, G2. Alright. And then, how do these phases link to each other? Happy? If you're happy, you can pass the marker back to me. Yeah? Okay, give Marsha a round of applause. Okay, wait, let me just take ribbons, pendants. Okay, the cell cycle. Yeah? Michelle drew the cell cycle on the board. In your OneNote, you may find that the cell cycle is represented a little like a pie. Seen that before, the diagram? Okay? You may see that the cell cycle sometimes is represented like a pie. But it's the same idea, that one phase kind of leads to the other. Okay? We call it a cycle because cells kind of go through these phases. They go through a period of growth. They go through a period of okay, so let's fill this in up. What happens at each phase of the cell cycle? Who's in there? Okay, Megan. Okay, first phase, G1, we often refer it as the gap phase 1. But actually, I see the G and then I think of growth. Okay? So the cell needs to grow first in G1. What is the S phase? The S stands for synthesis. Synthesis of what?
DNA.
Yeah, synthesis of DNA. More properly, we often refer to this, what's DNA? Okay, we often refer to this as DNA replication. To replicate something is to create a copy of. Okay, DNA replication. Last but not least, it goes through another G2 phase, and in this phase, you expect more growth and also synthesis of proteins. Finally, we go into the M phase, right? I'd like you to know that the M phase consists of many parts. Okay, you have your OneNote with you? Yeah? Okay, you have a look at the M phase. The M phase consists of several parts, right? If your notes look something like that, they put the M as a very small segment, and then they branch off to many parts, right? And then after that, over here there is a cytokinesis. You see that? Yeah? Okay. So the M phase consists of, if you look at a textbook, the M phase is when the cell divides. Okay? But I want you to be very specific about it. I'm going to direct you now. These many phases is when the nucleus divides. Cytokinesis is when the cell divides. What's the difference? So the difference is something like this. Imagine now I have a cell with a nucleus. Actually, we need the nucleus to be 2 first. Then we divide the cell into 2. Yeah? So, nucleus needs to divide first, then the cell divides. Okay? But in general, during the M phase, things are dividing into 2. Right? What's your Alright? Yeah. Cytokinesis. C-Y-T-O-K-I-N-E-S-I-S. Okay? Cyto means like cytoplasm. Kinesis means to split into 2. So we are splitting kind of the cytoplasm. Yeah? For those that are done, I would like you to think about this, or maybe Ms. Lim went through. Why does a cell need to grow and replicate its DNA before it divides into 2? Why not just like go through many rounds of M and M and M and M? Why does it need to first go through G, growing, DNA replication, and grow again?
For the DNA part, like if the nucleus just divides, then you only have half the amount of DNA required. So
Yeah, right? If you just go and chop the nucleus into half, then each cell only has half the DNA. Right? DNA is so critical. It's like it contains all the information to run the cell. We just chop into half, right? That kind of makes sense. It's like saying, I have a library, I want to create a new library in a school. I just take half of the books from the library and then shift to the other side. Yeah? So yeah, that works, right? But it is not quite replicating the library. We want a full set of the books on the other side. Okay? Why must we grow the cell before we divide the cell into 2? Sorry, say again.
like the organism also grows bigger with the cell. So the cell itself doesn't grow, it still occupies the same space even after it divides.
Okay. So you are taking this in relation to the entire organism. Okay. So cell division occurs at a cellular level. It's true. When many cells divide, you will grow. Right? So sorry, may I understand your point? Are you trying to say that it needs to, because you are getting bigger, you need to also, the cell also needs to grow bigger to fill up the space, is it?
No, like if the cell doesn't grow,
Yeah.
then you have 2 very small cells instead of
After you divide it.
normal size cells.
Okay. Does that make sense? Let's say I have a single cell, and then if it doesn't grow first, I chop into 2. Now I have 2 small cells. Go through another round of M phase, I will have even smaller cells. I'll just keep chopping and get smaller and smaller and smaller, right? So I hope you appreciate that there's some logic in why we have to go through these phases. We need to grow so that we don't get smaller and smaller cells. We need to replicate the DNA so that we don't get half the DNA each time. There is a point to all these phases. Okay? Collectively, we refer to these phases, G1S and G2S, the interphase. Kind of like the intermission. Right? You watch a play, then after that there will be an intermission where you can go and touch up yourself, put a makeup bag, drink some water, refresh yourself, then you go back into the main show, the movie. Okay? I'll take a pause right now for you to take down some notes. But this is all I'd like to go through for the cell cycle. Okay? Because the juicy part is the next part. And then I'll take questions, usually when I take a pause, yeah? So if you have any questions, now's the best time to bring it up. cabbage dog in front of you. It's not even a cabbage dog, it's a bok choy. It's a Chinese letters dog.
Chinese letters.
You know, this one is a Chinese one. This one used to make kimchi one, right? Oh, it's called tissue paper. Okay, everyone good? Okay, so just so that we are all very focused, from this chapter, okay, the next part I'm going to cover focuses only on this part, yeah? So you don't be all over the place. I'm only going to focus on the S phase right now. Okay. As a class and as GH4, we don't really focus on G1 or G2. Okay, I want you to focus on the S phase. This is the very important one. So today, right now, we're going to review what DNA replication is all about. Okay. So this checkbox is for you and for me, because when I come to class, I'll start chit-chatting with students and I forget what I'm doing. Okay, so this is for me also. Okay, so we just recap the cell cycle. Broadly, just understand that there are phases. There's a time and place for everything. For those that are done, you may go on to read the next part of the notes first. Okay, if you are done taking our notes, because I'm about to erase the board. Who was here for lessons last week? A lot of you all. Okay, so may I ask, right, did Miss Lim start on DNA replication?
He did a bit.
No. I just want to know whether she did or not. Did she?
She did a bit, like she told us to go Google search those two guys on the DNA.
Okay. Okay, so you all learn about how DNA replicates. But do you all see all the test tube thingies? You all did?
But she actually like two different
Okay, it doesn't mean Miss Lim go through already, then I won't go through. I just want to know if you understand or not. Because if you don't understand, then that's why I'm with you. Okay, so now let's just zoom in on the S phase. Okay, if you give me a while, I'm going to draw some diagrams on the whiteboard. Okay, now let's focus on the S phase and specifically DNA replication. Okay, this part I find over the years of teaching that some students find this part a little bit challenging. So if you have not been, haven't fully wake up, now's the time to really wake up. Just slap yourself twice. Okay, okay, I'll try to slap you. Ayo, I'm recording it. Yeah? Hey, I already heard a slap. You slap yourself or someone slap you? Okay. Right. Now, before we begin, can we recap the structure of DNA? Share with me. Yeah, what? Oh, okay. A double helix. Okay. Number one, DNA is made up of a double helix. Anything else about DNA? Okay, I'm sure that there are sugar phosphate backbone, and then I also heard that there are nucleotides. You remember all these things from JH3? Okay, anything else? Oh, okay, I heard ATGC. And then actually between ATGC you have complementary base pairing. All still coming back to you? Okay. So that means to say, the key thing we're going to talk about today is DNA replication. So you must understand the structure of DNA. So earlier on, okay, you shared that there are nucleotides, right? Okay, I hope these structures seem familiar to you. This is a simplified structure. Okay, those are the structures. Number one, we know that each nucleotide is made up of a sugar. Okay, that's all the hexagon parts. We also know the nucleotides have ATGC, right? Another word for these ATGCs, right, is nitrogenous bases. Do you know why they are called nitrogenous bases? Hey, very good guess. Okay, so now you go and zoom into your notes or OneNote. Look at the structure of the DNA. You look at the ATCG. Do you see nitrogenous? Do you see that there's nitrogen in the nitrogenous bases? Okay? Have not? Yeah? I need to be careful not to open the exam folder. Yeah? So, key thing, there is nitrogen inside these nitrogenous bases that make up DNA. Don't believe? You zoom in and have a look. Look at the ATCG. Do you see a lot of nitrogen inside? Yeah, that's why they're called nitrogenous bases. And then by now, right, you've learned that you've learned acids and bases in chemistry. Yeah. So, nitrogenous bases. So actually, all these things, they are bases. Can you find the base? Do you see the N and then double H? That's a base. Yeah? So I hope perhaps you can see in a different light. We call it nitrogenous bases because they are bases, not acid bases, and they have nitrogen inside. Okay? Alright, so now we've just done a quick recap of DNA. We know there is a double helix, so it's made up of two strands. And in these two strands, we find a lot of ATCG, which are nitrogenous bases. Okay, so that's the first thing we need to recap. So now let's go into DNA replication. We know that DNA is made up of two strands. Today I'm going to represent the double helix with two magnets. Okay? Each magnet represents one strand. One strand, two strand, double helix. Okay? Emily, can you see the words? So what does it say? Yeah, okay, original. Okay, good. Okay, so it says original. I would like you to, the reason why I wrote original because these are the original strands. What do I mean by that? DNA is a double helix. Let's talk about DNA replication now. Let's say I want to create a new strand of DNA. Okay? There were three theories in the past of how DNA replicates. I'm just going to focus on two of them. Alright? So let's have a look. First method of DNA replication is that I make use of my original strand to synthesize a new strand. Okay, a new double helix. So the word new means this is the new double helix. Okay? So this is the first theory that scientists have in the past. They call this theory the conservative method of DNA replication. Why? Because the original DNA strand, double helix, is conserved. Okay, this doesn't quite make sense until I show you the contrasting theory. Okay, the contrasting theory goes like this. Imagine I have a double helix. This time, instead of using this to immediately generate a new double helix, what if we make use of each strand as a template of its own? So up, okay, so each one has a template, and then we use each one as a template to create a new strand. Now, each of the new double helix is made up of one original strand and one new strand. This is a contrasting theory, and we call this theory the semi-conservative model of DNA replication. Semi-conservative because the new double helix is only semi-original. Half original, half new. Okay? These were the two theories. But we scientists in the past didn't know which one was true. And then came the two scientists that Miss Lima is going to Google about, Masterson and Stahl. Right? Masterson and Stahl came up with the most elegant and beautiful experiment ever in biology. At least that's what they call it, and that's what a lot of biologists call it. Because they managed to find out which theory holds true. Just through the understanding of the structure of DNA. Because they knew that DNA, nitrogenous bases is made up of nitrogen, they thought, what if I could visualize where these nitrogen go? So what did they do? They made use of an isotope of nitrogen. You know what isotopes are?
Yes.
Yeah? Isotopes. So what are isotopes?
Same number of protons and electrons, different number of neutrons.
Same number of P, different number of N. Yeah? So I give you an example of a nitrogen isotope. There's nitrogen 14, nitrogen 15. Which nitrogen is heavier? 15, huh? Then nitrogen 14 is the lighter one. Yeah? Okay, so for today, I'm going to label these nitrogen in different colors. Nitrogen 14 represents the nitrogen that's lighter, nitrogen 15 the heavier nitrogen. Okay, still following? So that means to say, if I were to grow a bunch of cells in nitrogen 15, their DNA will be really heavy, right? Yeah? So imagine now all the double helix really heavy. And it's an interesting part. If I were to take these cells, imagine all these are in bunch of cells, right? Then I go and kill them all, okay, like pop them, put them into a test tube and take it for a spin. Okay, something interesting happens. Okay, let's take it for a spin in a test tube. The DNA, because of its heavy weight, will all kind of sink to the bottom in one band. Yeah? Then if I were to take DNA, if I were to take cells and grow them in nitrogen 14 that's lighter, all the DNA will come out very light. Yeah? If I were to kill the cells and take them for a spin, you expect that the DNA, after spinning, right, it won't sink as low because they're lighter. Instead, it will end up somewhere at the top. Okay? This spinning process is a proper word for it. It's called centrifuge. Heard of this word before? So, yeah, maybe you've heard it before, centrifuge. Okay? Masterson and Stahl made use of just this knowledge to figure out which one actually holds true. I want to show you how beautiful it is. Okay? I'm not going to tell you the answer. I'd like you to figure it out for yourself, because if you do, then you will appreciate how beautiful their experiment is. Okay? So, this is what Masterson and Stahl did. Okay? They took a bunch of bacterial cells. Okay? They took a bunch of bacterial cells, and this is what they did. They grew them in nitrogen 15. So these bacterial cells are surrounded by nitrogen 15. As it's making its DNA. So, if I were to take these bacterial cells and I kill them all, and I take them for a spin, where would you expect the band of DNA to float up to? Lower? Kind of, if I were to match things up, it should be at the same height over here, yeah? Because I grew them in nitrogen 15, right? Okay, so all of the DNA will be pretty heavy. Okay? Okay, so this is the really elegant and beautiful part. This is what they did next. What if I take some of these bacterial cells and instead of growing them in nitrogen 15, they grew up in nitrogen 15. But now I change the environment. Now I grow them in nitrogen 14. Okay? Now I grow them in nitrogen 14. As a result, you expect to find DNA that is both heavy, but any new DNA form will be light. If I were to take these bacterial cells, I kill them, take them for a spin, I'd like you to discuss with your friends now. Where will you find the band of DNA? Okay? You must try this yourself. This is the part where you can figure out your DNA. Okay, here's a clue. When we take these cells for spin, the DNA doesn't break apart. The DNA will stay together as a double helix. Okay? They don't break apart when you take both for spin. They don't break apart. Ready? Okay. You see? I invite you up to draw where you expect to find the bands of DNA. Okay. Where will you find the bands of band or bands? I don't know. Okay. Is there one band? Is there two bands? Or three bands? Where will you find the DNA float up to if I take these cells for a spin? Okay, immediately she went for down below, and then there's some on top. Thank you, Izzy. Round of applause for her. Anyone has alternatives? No. Could be in the middle. Okay, could it be in the middle? Okay, so imagine we take these cells for spin, we kill the cells, we take the DNA for spin, right? You'll find all the heavy ones sink to the bottom. Good. Oh, really sink to the bottom. And then you find all the light DNA float to the top. Yeah? So having two bands kind of makes sense. Yeah? Okay, so now I think some of you already have thought ahead. Now let's look at this model. Okay? If I grow the bacterial cells again, starting off, okay, they grow up in N15. I take them for a spin, you will find that all the DNA will sink to the bottom again. Yeah? Okay, so here's the elegant part. I don't know if you can see yet. Imagine I take this generation, this is the first gen, second gen of bacterial cells, and let them grow in N14. Okay? The original DNA will all be heavy, but any new DNA that's formed will be light. So now I have double-stranded DNA that has both light and heavy. So this is the last part of the puzzle. What would you expect to see the DNA band if I were to kill these cells and take it for a spin? I would like everyone to try and think this through. Okay, you can solve this and you understand this experiment. And then I hope you can see how beautiful and elegant this experiment is. So simple. Okay, ready? Then I'm going to call someone to come and draw the band or bands. Ready? Ready? Ryan, can? Alright, come Ryan. Hey, yeah, Ryan, my big guy. Don't know why all this while I thought you were an essay student. Okay. Okay, where will Ryan draw the band or bands? Ryan has chosen to draw the band, just one band in the middle. Oh, is it two band or one band? Oh, okay, one for blue, one for red. Okay, the DNA doesn't split, it stays together. So when you take them for a spin, right, they don't like split up. Yeah, they stay together as a double helix. So there will be two of the same kind of. So, yeah. Is this two bands or one band? So there's one band on top, one band below. Oh, okay. Why do you draw them in the middle and not like up here and up here? One will drop down, one will go up. Okay, can you show me what I mean by that? Oh, then this one? I see. Okay, but what if they can float any way they want? Right? Right? Actually, they are exactly the same thing, right? No matter how you look at it, just from different angles. So actually, right, Ryan is correct that you'll find the DNA in the middle. Okay? But actually, it's just one single band. Yeah? It's just one single band. You'll find all the DNA in the middle. Okay? Floating in the middle. Yeah? Because they are half heavy, half light. Yeah? Okay, thank you for identifying this in the middle. Okay, thanks, Ryan. Okay, understand how it goes? Okay, so quite elegant experiment, right? All they did was to grow the bacteria in KB, and then second generation grow in light, to find out which theory holds true. So which one actually holds true? Yeah, so when they conducted the actual experiment, they realized they saw only this. Yeah? They saw only one band. And just by seeing only one band in the second gen, they know that it's semi-conservative. Yeah? And if they see two bands, then they know it's going to be conservative. Okay? So now, let's stretch your thinking a little bit further. If I were to grow it for a third generation, okay, if I grow it for a third generation, can you tell me what the bending pattern will be if I grow it for a third generation? So now this is a thinking puzzle. If I grow them in the third generation, where will you find all the bands? Okay, everyone think up. Where would you find the bands if I grow this for third generation? What do I mean by that? So there will be the original strand, and then there will be this strand over here. But any new DNA will be right. Any new DNA will be light. Okay. And what's the bending pattern here? Yeah, so this is quite a teaser, right? These are the original strands. This is also another strand. Okay, these are all the templates now. And I have 4 more over here that's light. To make new ones. So a new one, new one. So what's the bending pattern for both? When you go to the third gen? Okay, anyone like to volunteer this time? I'm sure marking your face. Okay? Ready up? Yuan, you can? Yuan, you want to choose which one you want to do? This one or this one? This one? Okay, come Yuan, thank you. Okay, you will attempt to draw the bending pattern for the third gen in conservative. What would it look like? How many bands will you see? Yuan has drawn a band at the bottom again. He's drawn a band at the top. Okay? Yeah? Is there a difference in this? Okay. Yuan has drawn a band on top that's really thick. And then the band below the same as the second gen. Why do you draw this band to be so thick? Because there's more light DNA, right? Okay. Thank you, Yuan. Okay, thank you, round of applause. Okay, then this one how? What would the bending pattern look like? Kiran? Now Kiran has been like actively answering, so I get you to write the board. Thank you, Kiran. Hey, quick. Then just come out without your shoe lah. Okay, thank you. So you draw the bending pattern for me, ah. So he drew a one. Okay, wait, I don't understand. Is this one band by itself? This is one, this one.
But there are 4 bands there.
Wait, where are the bands?
You have to scribble. Wait, scribble.
Oh, okay. So Kira is saying this is one band. Is this the middle? You drew in the middle, is it? Then this is another band on top, is it? Okay, can. Okay. Okay, now your band became thinner from this side.
Oh no, it's not based on the previous.
Okay, it's not based on previous one. Can. Okay, so the idea here is that you have a band in the middle and a band on top. Will the thickness be the same? Yeah, thickness should be the same. Yeah. Okay. So, okay with this experiment. Yeah. Students often find this really tough, but I hope you find it okay. 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 So in the event that I was not here, yeah. Oh, you can expect to find a slightly thicker band. Okay? Yeah. Because at the end of the day, I'm not working with just 4 DNA. I'm working with a lot of cells. Okay, in the event I was not here, you know I have a website called Lumos. Okay? If I was not here, I'd like you to go to Lumos because you can find videos to help your learning. Every chapter you have learned or will learn is here. For example, you can go to cell division, which is this one over here. Under cell division, you can find videos that teach you about the cell cycle. You can also find a video of me teaching this same thing. Okay? So I want you to know that you are not alone. Okay? That you can always catch up. And if you don't want to listen to me teach, guess what? The 2 people that came up with this experiment taught the experiment themselves. Yeah, Masterson and Stahl. Yeah, I got a shock, you think that Masterson and Stahl by now, right, already like pass on. Boy, they're just 2 really old dudes. And this is Masterson himself, Matthew Masterson, teaching his own experiment. Wow, this one I was like, okay. So, if you want to get to know them better, you can go down and you can find Masterson and Stahl. Okay? The most beautiful experiment in biology. Okay? Okay, I just let you all watch this, right? So that you can let's put a face to them, The 2 amazing scientists. Right, so they can see what they look like also. Okay, this is what Masterson and Stahl look like. Yeah, this is them now. Oh no, this is Watson and Craig. This is Masterson and Stahl. Hey, when you reach this age, right, you also want to wear shorts. So cooling. Yeah, okay, so this is them. Just wanted to show you what they look like. So it's a very nice short documentary. Okay? Alright, so today what we spent our time doing was DNA replication. This is my homework to you. Can you therefore complete the SLS? Okay? The SLS has a few more puzzles for you to try to solve. Solve the puzzles to see if you understand what has been going on. Right? That's what's happening for the last part of SLS. After solving a puzzle, you will find just one reflection at the very end to do. Okay? When can you complete the SLS by? I need to know a deadline so that I can keep track. Ready? So, really, honestly, when is the fair time for this to be completed? The next lesson is tomorrow. Is it fair to expect this SLS to be completed by this Friday? Okay? Okay, so by this Friday, when I go into the SLS, all should be completed. Fair? I will check. And then after that, I will keep track. If you cannot complete, you let me know. Okay? Tomorrow's lesson, we will be going into the lab to use microscopes. Who here cannot use a microscope? Who's not very confident in using a microscope? Okay, who is like a boss? You go in, right? You just try a microscope. Put anything under you can find stuff. Yeah, I don't have that boss. Yeah, I'm the boss. Okay, so tomorrow we'll go and use the microscope. What's the purpose of your garlic? Okay, I want you to go home and check. Your garlic should have sprouted roots by now. Have not? Okay, I don't have. Okay, if you don't have, right, then tomorrow you need to You need to share roots from other people. Okay? If you find roots, this is what we'll do tomorrow. Those roots that are really short are the ones that we're going to slip off. Okay? Because the roots that are really short are the roots that are actively still growing a lot. Okay? This whole chapter is about cell division. We want to see what cell division looks like. So we're going to chop off the roots that are actively still dividing. Okay? And then what we'll do next is that we will take the roots, we will stain it a blue color, and then we'll put it under the microscope to try to find cells that are dividing. Okay? That's the aim of tomorrow's experiment. So you must come with roots. Or no, the garlic, chop off. Don't chop first, come to class then you chop. But I need you to do a little bit of reading first. Okay? I need you to go to, okay, anyway, today we covered this. We did page 3, did page 4, did page 5. For tomorrow's experiment, I'll need you to jump all the way to here. I'd like you to go to page 8 of your textbook. Okay, page 8 of your textbook. This is your homework for tomorrow. Some must be by tomorrow. Page 8. I need you to read page 8. Memorize what the different phases of the dividing process looks like. Because tomorrow, under the microscope, I want you to find all these phases. Okay? Yeah, you must read page 8 by tomorrow. Okay? Earlier on I was saying that when a cell divides, the nucleus needs to divide first, right? Okay? There is a name for this. Okay? The process when the nucleus divides is called mitosis. Okay? Tomorrow, our aim is to look under the microscope and to find cells that are undergoing mitosis. Mitosis has a few phases, as you will read on this page. There is a prophase, there is a metaphase, there is a anaphase, there is a telophase. Okay, there are 4 phases. TMAT. Okay, TMAT. Today, I need you to read up on these phases. Memorize, right, visually what these phases look like. Because tomorrow, when we look under the microscope, I want you to see if you can spot these phases. Okay? And if you didn't read this up tomorrow, when you look at the cells, they just look like a mess to you. Okay? Then there's no point of the practical. I would prefer you to remember what it looks like that. Because this is the microscope view. This is a picture view. Okay? So if you memorize this one, high chance you won't find it. Okay? You won't see red and blue sausages floating around. Okay? But you may see a mass of chromosomes in the middle, then looking like they're moving apart. This is what you will actually see. Okay? So remember this clearly. What should you bring tomorrow? Please bring your iPad along into the lab cat other than the garlic. Because if you cannot spot it, then you can take out your OneNote, look at your notes, and then you try to spot. Okay? And then we see how many you can spot tomorrow. Okay? On top of that, I want you to learn how to use a microscope fast. Okay? What thing happening? Oh, okay. So, will you see the cells divide before your very eyes? I would say maybe yes, but you cannot tell because it's happening pretty slowly. So it may be happening, but you cannot tell, it's too slow. Unless you stare there for 1 hour, right? Then maybe you will see some change. Or you take a time lapse, maybe you will see a change. Okay? Then bring your phones along because I want you to take beautiful photos of what you see. Okay? Enough? Okay. Okay, any questions? That's all I have for today. Please spend some time now to organize your OneNote. Take down notes that you haven't taken down. Or you can work on your SLS now. Yeah? Today now we must sing song, right? Yes, Marcus? very loud one. Yeah.
It's not a state of mitosis, but it's part of the phase. So M phase does not only include mitosis, it includes mitosis plus meiosis. So that's why it's okay to say that even also like yeah. So cytokinesis is not mitosis, but both of them are under mitosis. Yeah, right. So this type of maybe one of them.
Ratio of light to heavy is 1:1. Ratio of light to heavy here is 3:1. So based on the pattern you see there, what is the ratio? Like this one? Oh, what's the ratio? Is it? Oh, the ratio is just. There is no ratio. Sorry? That is semi-heavy. So it's just semi-heavy. There is no ratio to play with. Unless you really want to say semi-heavy to heavy is 1:0. Semi-heavy to light is 1:0. There's only this. Will your form teacher not be coming down to sing national anthem? No, so I'm going to sing with you. Lovely, may you stop the